Hello, friends. Welcome to ENTRANCEADVISER.com. Today, I'll be continuing with the topic reproduction in flowering plants. Uh, the topics are double fertilization, post fertilization structures and events, apomixis, and polyembryony. Before that, if you are new to my channel, then please hit the bell icon and do subscribe to my channel. So, let's start. What is pollination? Pollination is the act of transferring pollen grains from the male anther of a flower to the stigma of a female plant. The goal of every living organism, including plants, is to create offspring for the next generation. One of the ways that the plant can produce offspring is by making seeds. Seeds contain the genetic information to produce a new plant. We call the animals or the insects that transfer the pollen from plant to plant are called pollinators. Here you can see the butterfly and the honeybee are the pollinators who are transferring the pollen grains. So, pollination is an ecological process carried out by all flowering plants. In this process, the matured pollen grains are transferred from the anther to the stigma of the purpose of sexual reproduction in the flowering plants. There are two types of pollination. One is self, one is cross. In the self, the process occurs when the pollen grains from the anther to the stigma are deposited on the same flower or another flower of the same plant. In case of cross pollination, the process occurs when the pollen grains are transferred from the anther of one flower into the stigma of another flower of the different plants of the same species. So, in these pictures, you can clearly see that the same pollen grains are transferred in the same plant. The anthers are produced, which contain the male gametes, are deposited on the same flower or the same plant. And in case of cross pollination, you can see one agent is performing, and the pollens from the stamen sticks to a bee as it visits a flower to collect food. The pollen or the bee sticks to a pistil of a flower on the another plant is done. The pollination. As we can see from the above discussion that it is a transfer of pollen grains from anther to stigma in simple words. So there are several methods or techniques which plants follow to do the pollination. Those are autogamy, claistogamy, and chasmogamous. Autogamy is divided into two groups. The transfer of pollen grains from the anther to stigma of the same flower. The clastogamous means the flower which is do not open, and clastogamous flowers are autogamous, as there is no chance of cross pollen landing on the stigma. Clastogamous flowers produce associated seed FN in the absence of pollinators. Example, the viola, the common pansy, oxalis, and camellia. The chasmogamous flowers are exposed anther and stigma. That means they are open. Second one is geitonogamy, the transfer of pollen grains from anther to stigma of the different flower of the same plant. The geitonogamy is functionally cross-pollination involving the pollinating agent. Genetically, it is similar to autogamy since the pollen grains come from the same plant. And xenogamy is the transfer of the pollen grain from the anther to stigma of the different plants flowers of the same species. Here are several pictures which clearly depicts that the cross pollination occurs and the self pollination is known as autogamy, and this is also self pollination but it is geitonogamy. Only the differences is the flowers are open, and the chasmogamous the flowers are closed, and in the geitonogamy it is also the transfer from anther to stigma of the different flower of the same plant. As I said, the self pollination can occur in two ways. And in case of xenogamy, it is not shown clearly, but here it is there. The insect is doing from one plant, it is transferred to another plant, and this is autogamy, and this is geitonogamy. This picture is good. Now the agents of pollination. There are various agents. Abiotic such as water and wind, and biotic insects such as butterfly, honeybee, etc. Large number of pollen grains are produced by the plants using a biotic mode of pollination, as most of the pollen grains are wasted during the transfer. This is a wind pollination, this is water pollination. Let's come to the text. The pollen grains are light and non-sticky and winged, anther well exposed, stigma large and feathery, flower one ovule arranged as inflorescence, example cornscob, cotton, date palm, etc. The water pollination are beared by bryophytes, pteridophytes, algae, etc. The pollen grains are protected by mucilaginous covering. The freshwater plants Vacciniaria, Hydralia, and seagrass Zostera. The main features of the wind and water pollinated plants are produce a large number of pollen grains as most of them are get wasted and do not produce nectar. In case of insect pollination, they produce the nectar, as you know, the large, colorful, fragrant, rich in nectar flowers. And the pollen grains are sticky, stigma is sticky, so that they can stick to the legs or the feet of the insect and it is easily transferred. Certain rewards to the pollinators. Nectars and edible pollen grains as food provides the safe place for laying eggs, Amorphous and Yucca. The outbreeding devices are the various mechanisms taken to discourage the self-pollination and encourage the cross-pollination. As continued, the self-pollination leads to the inbreeding depression. It includes the pollen release and stigma receptivity not synchronized. Anther and stigma are placed at different positions, inhibiting the pollen germination in pistil, production of unisexual flower. So, what is inbreeding depression? When the self pollination occurs from generation after generation, the variation or the gene pool is lost, or there is no variety occurs. So, the repetition of genes is increased and it leads to inbreeding depression. And pollen pistil interaction is the pistil has the ability to recognize the compatible pollen to initiate the pollen pollination events that leads to the fertilization. The pollen grain produce pollen tube through the germ pores to facilitate transfer of male gametes to embryo sac. What is artificial hybridization? The crossing of different varieties of species hybrid individual with desirable characters of the parent plants. The desired pollen grains for the pollination are stigma protected from the contamination. It is a very much important. What is emasculation? It is the removal of an anther by the help of a forceps. And what is bagging? It is an artificial process. Those are artificial process only. Bagging is a flowered covered bag made up of butter prevent for the contamination of the stigma from the unwanted pollen. The bag flower attains receptivity, mature pollen grains dusted on the stigma, rebagged and fruits allowed to develop. Plastic bags are made made up of maybe butter paper also to prevent the external pollination. What is double fertilization? After entering one of the synergids, each pollen grain releases two male gametes. One male gamete fused with the egg syngamy and other male gametes fused with the two polar nuclei. Triple fusion occurs to produce the triploid primary endosperm nucleus, which is known as PEN. Since two types of fusion takes place in an embryo sac, the phenomenon is called the double fertilization. The pen develops into the embryo and zygote develops into embryo. Endosperm and zygote develop into embryo. So first of all, what happens? There are two antibodal cells. Antibodals, the polar nuclei synergids are present, and the release of sperm cell occurs, and the 3n number of endosperm produces, and then it develops into an embryo. The first of all, the PN develops into an endosperm. Then the zygote develops into an embryo. The two different development occurs or the fertilization events occur. So this is known as double fertilization. This is a detailed view. You can see the pollen grains adheres to the stigma which contains two cells, a generative cell and a tube cell. The pollen tube grows into a style. The generative cell travels inside the pollen tube. It divides to form two sperm. The pollen tube penetrates an opening in the ovule called a micropyle. Then one of the sperm fertilizes the egg to form the diploid zygote. The other sperm fertilizes two polar nuclei to form the triploid endosperm, which will become a food source for the growing embryo. In this picture, as you can see, it is a fertilized embryo sac showing in the zygote and primary endosperm nucleus PEN. The stages in the embryo development in the digote is shown as a reduced size as compared to picture A. Now, the post-fertilization events include the endosperm and embryo development, maturation of ovules into seeds and ovary into fruits. The endosperm. The primary endosperm, the cell divides many times to form the triploid endosperm tissue having the reserved food materials. The two types of embryo development are the free nuclear type and the cellular type, which is further divided into two groups. Non-albuminous endosperm completely utilized before maturation seeds, example pea and groundnut. Albuminous, a portion of the endosperm remain in the mature seeds, example wheat, maize and castor. You know what is endosperm is. This is very much familiar. You are very much familiar with this word from the very much childhood. This is a nor kernel and this is whole. This is seed coat. This is endosperm and this is a embryo. And this is a cross-sectional view. Now what is embryo? Embryo develops at the micropylar end of the embryo sac where the zygote is located. Here, the embryogeny, early stages of embryo development, the zygote gives rise to the proembryo and subsequently to the globular, heart-shaped and mature embryo. The embryo consists of following parts: embryonal axis, cotyledon, spinule and radicle. The monocotyledonous seeds contain one cotyledon and scutellum means the cotyledon, coleorrhizia, the undifferentiated seed covering the radicle and root cap, and coleoptile, the seed covering the plumule. This is the seed coat. This is a fruit wall. This is aleurone layer. This is the endosperm. This is scutellum. This is coleoptile. This is plumule. This is radicle, and this is coleorrhiza. Here, the stages of embryo development of a dicot embryo is shown as shown in the previous picture. You may refer also to this also. Then the seed. The fertilized and mature ovule develops into the seed. The seed consists of cotyledons, the embryonal axis. The seed coat, the double layered, formed by the integuments, the testa, outer coat, tegment, the inner coat, the micropyle, the small opening on seed coat. It facilitates entry of H2O and oxygen into the seeds for germination. Hilum, the scar on the seed coat. The albuminous or the non-albuminous peristome, the remnants of the nucleus that is persistent, example black pepper. Here is the structure of the seeds in the dicots and monocots, the bean and the wheat. Bean is a dicot plant and wheat is a monocot plant. It is a dormant seed. And what is a dormant seed? The state of inactivity. The wall of the ovary develops into the wall of the fruit called the pericarp. In the true fruits, only ovary contributes in fruit formation, by in the false fruit thalamus also contributes in the fruit formation. What is apomixis? Form the asexual reproduction mimics the sexual reproduction seed formed without the fertilization. Formation of apomictic seeds, the diploid cells formed without the meiosis, develop from an embryo but without any fertilization. The cells of the nucleus are two ends surrounding the embryo sac protrude into the embryo sac, develop into the embryos. Example, the citrus and the mango. These are the few examples which you can refer for the apomictic diagram. Here the seed is developed, fertilization occurs, known as pseudogamy, and without any fertilization. It's a very much complex process and all. And what is polyembryony? Occurrence of more than one embryo in a seed. There are multiple embryos, often associated with the apomixis, example citrus and groundnut. This is a image. I know this image is not clear here, but this is also visible also, and degenerative pollen tube is there, and this is embryo sac, and among this the development of embryo from the antipodal cells occurs, and the pre-embryos are developed from the cell's nucleus as there are multiple embryo you can see here at last. So thank you for listening and take care. Bye bye.